Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Continuing Education, SOCE, Master's Degree Programs, Master of Science, Food Nutrition, MSCDFSM, Revised First Year MFN 002 Nutritional Biochemistry, unit-4 Enzymes and Coenzymes 4.1 Introduction. Enzymes and coenzymes are two of the most important groups of biomolecules, without whose active participation, none of the nutrients discussed in the last 3 units can be utilized by the body. As a learner of dietetics, it is therefore essential for you to get the knowledge of different structural and functional aspects of enzymes and coenzymes. This unit focuses on these aspects, which will enable you to understand the metabolism of different nutrients, which are to be discussed in subsequent units of this course. Remember, this unit along with the next 3 units forms the basic component of this course. Objectives: after studying this unit, you will be able to classify enzymes and coenzymes, discuss their chemical nature and functions, describe the factors on which enzyme activity depends, and explain the diagnostic use of enzymes. 4.2 Introduction to Enzymes and Coenzymes. Let us start by understanding what enzymes are. Enzymes are the proteins that catalyze biochemical reactions. Study of these important biochemical reactions was started many years ago, from the time of Louis Pasteur, who for the first time demonstrated the fermentation of glucose by yeast. The catalytic agent of yeast cell was subsequently identified and named as ferment. At the end of 19th century, a cell-free extract of yeast was found to be capable of fermenting glucose by Buckner brothers. Considerable advances were made since then in order to properly know the nature of these agents named as enzymes, and finally their true nature was revealed by James Summer in 1926 after the extraction and crystallization of the enzyme urease from jack beans. At present, no less than 150 enzymes have been prepared in crystalline form. What is the chemical nature of enzymes? Let's find out. We all know that enzymes are proteins. Do you recall the structure of proteins described in Unit 2 earlier? Enzymes are high molecular weight compounds made up principally of chains of amino acids linked together by peptide bonds, as shown in Figure 4.1. An enzyme can be a large protein made up of several hundred amino acids or several polypeptides that act together as a unit. Enzymes have molecular weights ranging from 10,000 to 2 million. Figure 4.1. Protein structure. Until recently, it was understood that all enzymes are proteins. However, observations made in organelles from plants, yeast, viruses, and higher eukaryotic cells show that RNA can act as an enzyme. Such RNA molecule is called a ribozyme. Hence, ribozymes are RNA molecules with catalytic activity. These generally involve transesterification reactions, and most are concerned with RNA metabolism, splicing, and endoribonuclease. Recently, a ribosomal RNA component was noted to hydrolyze an aminoacyl ester and thus to play a central role in peptide bond function, i.e. having peptide L-transferase activity. Before we move on to understanding more about enzymes, we should familiarize with the vocabulary and definitions of various terms which you will come across while studying about enzymes. Definitions of terms related to enzymes: cofactor, organic molecules or icons that assist many enzymes in their reactions. Apoenzyme, the protein portion of an enzyme requiring a cofactor for its reaction. Haloenzyme, a whole enzyme, as a complete and functional molecule. Generally, a holoenzyme consists of a polypeptide portion, an apoenzyme, and at least one cofactor or another coenzyme. Enzyme proteins that act as catalysts, speeding the rate at which biochemical reactions proceed but not altering the direction or nature of the reactions. Coenzyme, an organic, non-protein molecule that binds with an apoenzyme, a protein molecule, to form an active enzyme. Coenzymes are often derived from vitamins. Endoenzyme, an enzyme which is not secreted or exported out of the cell, but is kept and used by the cell which made it. Substrate, the specific molecule an enzyme acts upon. Metabolism, all of the organized chemical reactions in a cell which are under the control of enzymes. Catabolic reactions, reactions in which chemical compounds are broken down. Anabolic reactions, reactions in which chemical compounds are synthesized. Enzyme immobilization, the attachment of an enzyme to a solid matrix so that it cannot escape but can still act on its substrate. Enzyme inactivation, the disappearance of activity of an enzyme, in vivo or in vitro, due to presence of inhibitor molecules or inhibitory conditions, changes in pH, temperature, salt concentration, etc. Enzyme kinetics, quantitative characteristics of enzymatic reactions. Enzyme stabilization, reducing the chances that an enzyme will inactivate, in vivo or in vitro, by changing the environmental conditions, such as pH, temperature, concentration of salt, etc., or by attaching organic groups to it or changing some of its amino acid subunits. Having learnt about the various terminologies related to enzymes, let us now continue with our study on enzymes. Enzymes are synthesized within the cells and sometimes can pass through cell membranes under certain conditions. Enzymes which act inside the cell are called as endoenzymes, and those which are liberated outside the cell are called as exoenzymes. While enzymes like those involved in the production of energy belong to the first category, digestive enzymes belong to the second category. Enzymes, though basically are proteins, may sometimes have a non-protein component attached to the protein part. This may be an organic compound or a metal ion. While the first is called as coenzyme, the second is best known as cofactor. The protein part and non-protein part together form the holoenzyme, as illustrated in figure 4.2. Sometimes, the non-protein part remains so tightly bound to the protein part that it cannot be dissociated. Such non-protein part is called as a prosthetic group. When the non-protein component dissociates from the protein part, the enzyme loses its catalytic function and is called an apoenzyme. This, coenzymes, which are usually vitamins, sometimes form an integral part of the active enzyme and make them indispensable for the enzyme to carry out the catalysis. You have already learnt about the classification of vitamins in the last unit and know that these are broadly classified as water-soluble and fat-soluble. Remember that water-soluble and not fat-soluble vitamins constitute the precursors of coenzymes. While participating in the mechanism of catalysis, a coenzyme undergoes alteration and its restoration requires the involvement of another enzyme. As coenzymes may participate in a variety of reactions catalyzed by different enzymes, it is always convenient to classify them on the basis of nature of element or group such as hydrogen, carboxyl, amino, etc. they transfer in these reactions. We shall learn more about the classification of enzymes in the next section. 4.3 Nomenclature and classification of enzymes. Enzymes are generally named by the addition of S to the root, indicating the substrate on which the enzyme acts. For instance, the enzyme which catalyzes the conversion of maltose into glucose is called as maltase. Many common names such as aldolase also persist, which do not tell about the substrates, although the type of reaction can be recognized. Besides, trivial names of many enzymes are in use which do not follow this convention at all, e.g. pepsin. In order to follow a systematic nomenclature for all the enzymes and to classify them, the International Union of Biochemistry, IUB, has established a system whereby the enzymes are placed into one of the 6 major classes as summarized in Table 4.1. Each class is then subdivided into several classes, which are further subdivided. The system for classification of enzymes also serves as a basis for assigning code numbers to them. The code numbers are prefixed by EC, Enzyme Commission, and contain 4 numbers separated by points, with the following meaning. A. The first number shows to which of the 6 main classes an enzyme belongs. B. The second figure indicates the subclass. C. The third figure gives the sub-subclass. And D. The fourth figure is the serial number of the enzyme in its subclass. So you can see that a number is assigned to each class, subclass and sub-subclass so that an enzyme gets a 4-digit number. The 4th digit, in fact, identifies a specific enzyme. For example, alcohol, NAD oxidoreductase is assigned the number 1.1.1.1 because it is an oxidoreductase, the electron donor is an alcohol and the acceptor is the coenzyme NAD. Thus, in naming an enzyme, the substrate is stated first followed by the reaction type. The trivial name of the enzyme is alcohol dehydrogenase. Similarly, the EC number of catalase is AIC1.11.1.6. The first digit, 1, indicates that the enzyme belongs to oxidoreductase, class 1. EC 1.1 indicated subsequent digits representing subclasses and sub-subclasses. Major classes of enzymes and the types of reaction catalyzed by them are summarized in Table 4.1. Table 4.1, Major classes of enzymes and the types of reaction catalyzed by them. Look at the screen from the classification above. The different classes of enzymes identified include oxidoreductases, transferases, hydrolases, lyases, isomerases and ligases. Let's get to know more about them and their nomenclature. Oxidoreductases. To this group, EC1, belongs all enzymes catalyzing oxidation-reduction reactions. Common names include dehydrogenases, oxidases, reductases and catalases. The substrate, R2, that is oxidized is regarded as a hydrogen donor, AH2 plus B equals A plus BH2. The recommended name is dehydrogenase, but, as an alternative, the term reductase is used. The name oxidase is restricted to enzymes which exclusively use O2 as the hydrogen acceptor. The second figure in the code number of oxidoreductases indicates the group in the hydrogen donor which undergoes oxidation, e.g. CHO, CHO, CHCH, CHNH2, NAD, pH. Therefore, AIC 1.1 indicated acting on the CHO group of donors. AIC 1.2 acting on the aldehyde or oxo group of donors, etc. The third figure indicates the type of acceptor involved. 1 denotes NAD, P, 2 a cytochrome, 3 O2, 4 a disulfide, 5 a guanine, etc. So, AIC 1.1.1 states with NAD plus or NADP plus as acceptor, AIC 1.1.2 with cytochrome as acceptor, and so on. The list goes on. Transferases. These are the enzymes, code AIC2, which catalyze the transfer of a group, e.g. a methyl or glycosyl group, from one compound to another. In many cases, the donor is a cofactor, coenzyme, carrying the group to be transferred. Common names include acetyltransferase, methylase, protein kinase and polymerase. The second figure, EC 2.1, in the code number of transferase indicates the group transferred, a carbon group, 2.1, a carbonyl group, aldehyde or ketone, 2.2, a glycosyl group, 2.3, and so on. The third figure informs of the group transferred, e.g. subclass 2.1 is subdivided into methyltransferase, 2.1.1, hydroxymethyl and formyltransferase, 2.1.2, and so on. Hydrolyses. These enzymes, code AIC3, catalyze the hydrolytic cleavage of CO, CN, CC and some other bonds, including phosphoric anhydride bonds. Their trivial names are formed by adding the suffix -s to the substrate which they hydrolyze. Examples include protease, nuclease, phosphatase. A number of hydrolases acting on ester, glycosyl, peptide, amide or other bonds are known to catalyze not only the hydrolytic removal of a particular group from their substrates, but also the transfer of this group to a suitable acceptor molecule. Yet, they are not grouped as transferase because the transfer of a specific group to water as the acceptor molecule is considered to be their main physiological function. The second figure in the code number of hydrolases indicates the nature of the bond hydrolyzed, e.g. esterases, 3.1, glycosidases, 3.2, and so on. The third figure generally specifies the nature of the substrate, e.g. carboxylic esters, 3.1.1, thiol esters, 3.1.2, phosphoric monoesters, 3.1.3, O glycosides 3.2.1, N glycosides 3.2.2, and so on. Lyses. These enzymes, code IGNOU, cleave CC, CO, CN and other bonds by elimination, forming double bonds or conversely adding groups to double bonds. Common names include decarboxylase, aldolase, dehydratase, if water is eliminated, or hydrolyase, if the reverse reaction is more important or the only one which can be demonstrated. Synthase, but not synthetase, may be used as in tryptophan synthase or cystathionine beta synthase. The second figure in the IGNOU number indicates the bond being cleaved for e.g. CC lyses 4.1, CO lyses 4.2, and so on. The third figure informs about the group that is eliminated, e.g. CO2 4.1.1 or H2O 4.2.1. Isomerases. These enzymes, code IGNOU, catalyze geometric or structural changes within a molecule. According to the type of isomerism, they may be called racemases, epimerases, cis trans isomerases, 5.2 isomerases, tautomerases, mutases, or cycloisomerases. IGNOU 5.1 are racemases or epimerases. IGNOU 5.1.1 indicates acting on amino acids and derivatives, and so on. Ligases, synthetases. These enzymes, code IGNOU, catalyze the linkage of two molecules coupled with the hydrolytic breakdown of a pyrophosphate bond in ATP or an analogous compound. The bonds formed are often high-energy bonds. The second figure in the code number indicates the bond formed, e.g. CO 6.1, CS 6.2, etc. Examples include peptide synthase, aminoacyl tRNA synthetase, DNA ligase and RNA ligase. We hope the information given above may have helped you in understanding the classification of enzymes. The system of the nomenclature and the classification of enzymes are based exclusively on the reaction that is catalyzed and does not consider their origin or multiplicity. Enzymes catalyzing the same reaction, but isolated from different species, will have varying amino acid sequences so that they may be distinguished by electrophoretic methods. They may have different sizes and net negative charges and may even differ in their catalytic behavior. With this basic knowledge about enzymes, we shall go on to learn about the specificity of enzymes. 4.4 Specificity of enzymes. One of the major characteristics of enzymes is that they are highly specific. Enzymes, the organic catalysts, differ from the inorganic catalysts in their extraordinary specificity. Each of the enzymes that have been isolated and studied is found to possess different types of specificities. A few enzymes exhibit absolute specificity, that is, they will catalyze only one particular reaction. Other enzymes will be specific for a particular type of chemical bond or functional group. In general, there are 4 different types of specificity. These are as follows: reaction specificity, bond specificity, group specificity, optical specificity. We shall look at each of these specificities one by one. Reaction specificity I. Some enzymes catalyze only one reaction acting on a specific substrate. Example: urease and catalase acts only on urea and hydrogen peroxide, respectively. This is also called absolute specificity. E. Many enzymes can catalyze same type of reactions, phosphate transfer, oxidation reduction, hydrolysis etc., in several structurally related compounds. Example: carboxypeptidase acts on protein chains and removes one amino acid at a time from the C-terminal, irrespective of the nature of amino acid. E. A substrate can undergo many reactions, but in a specific reaction, an enzyme will catalyze only one of these reactions. Example: citrate synthase converts oxaloacetate to citrate in the presence of acetyl-CoA. But, in absence of acetyl-CoA, oxaloacetate is acted upon by a different enzyme, malate dehydrogenase, with the formation of malate. Bond specificity I. Some enzymes act on a particular bond, glycosidic, peptide, ester etc. Examples: pepsin, trypsin, chymotrypsin etc., are all only acting on peptide bonds, though the peptide bonds on which they will attack depend on the amino acids that have formed the bonds. Group specificity I. Some enzymes prefer a specific functional group to be present on the substrate molecules. Example: alcohol dehydrogenase acts on alcohols having O group. Some proteases act on the peptide bonds depending on the amino acids involved in the formation of the bonds. Example: chymotrypsin hydrolyzes only the peptide bond which is formed by the carboxyl group of phenylalanine, tyrosine or tryptophan. Optical specificity 1. Some enzymes exhibit absolute optical specificity for at least a portion of the substrate molecule. For example, the enzyme maltase catalyzes the hydrolysis of alpha-glycosidic bond between two glucose molecules but not the beta-glycosidic bond. Enzymes are able to discriminate between the optical isomers and act accordingly. Example: L-amino oxidase acts only on L-amino acids and not on D-amino acids. Having gone through the section above, it is clear that enzymes can be very specific. Next, let us learn about their mechanism of action. But first, let us recapitulate what we have learnt so far. So far we have learnt about enzymes, their chemical nature, classification and some characteristics. Next, let us learn about the mechanism of enzyme action. 4.5 Mechanism of enzyme action. L. Michaelis and M. L. Menton developed a general theory of enzyme action in 1913. According to Michaelis and Menton, the enzyme E first binds the substrate S to form a transient enzyme-substrate complex, S. This complex then dissociates into the product, P, and the unaltered enzyme, A. Let us understand how this S complex forms. The action of an enzyme is initiated when the reactants, i.e. substrates, bind at the catalytic sites or active sites on the enzyme molecule. The catalytic site of the enzyme molecule possesses a complex three-dimensional form and provides a cleft, which binds the substrate as shown herewith. A change in the tertiary or quaternary structure of the enzyme may alter the three-dimensional shape of the catalytic site and thus reducing its binding and catalytic activities. The S complex is formed mainly by non-covalent bonds between specific groups of the substrate molecules and the specific amino acid side chains present at the catalytic site of the enzyme. Different models for enzyme-substrate complex formation exist. Let us look at these models next. Models for enzyme-substrate S complex formation. There are two popular models to explain the enzyme-substrate interaction. These are Fisher's template or lock and key model and Koshland's induced fit model. The two models are described next. Fisher's template or lock and key model. According to this model, the catalytic site of the enzyme has a proper conformation compatible to a specific substrate even in the absence of the substrate molecule, as shown in figure 4.3. The catalytic site binds the substrate and catalyzes the reaction without any change in its own three-dimensional conformation. It has become possible to explain the specificity of many enzymes for only one of the stereoisomers of the substrate by this model. This model, however, failed to explain the change in enzyme activity in presence of allosteric modulators, low molecular weight regulatory substances that bind at a specific site on the enzyme molecule other than the catalytic site and thereby enhance or inhibit the enzyme activity, or the action of the non-competitive inhibitors, about which we will learn later in section 4.8 in this unit. Figure 4.3. Fisher's template or lock and key model and Koshland's induced fit model. Koshland's induced fit model. This model considers a flexibility in the three-dimensional conformation of the catalytic site. According to this model, despite having the required amino acids, the catalytic site of the enzyme does not possess the conformation complementary to the substrate in absence of the substrate molecule. Only when the substrate approaches towards the enzyme or during its binding, the conformation of the catalytic site changes so that the enzyme can hold the substrate properly, as shown in figure 4.4. This model, therefore, can suitably explain the non-competitive inhibition and allosteric modulation of the enzyme. We will learn about the non-competitive inhibition later in this unit in section 4.8. While on the topic of the mechanism of enzyme action, it is also important to know about the unit of enzyme activity, i.e., how to express the activity of an enzyme. Have you heard of the term kcal? Recommended unit of enzyme activity is called as kcal, which is the amount of an enzyme that transforms 1 mole of substrate into product in 1 second. In fact, other than kcal, the activity of an enzyme may be expressed in different other ways as highlighted herewith. International Enzyme Unit, U, is defined as the amount of enzyme that catalyzes the transformation of 1 mu more of substrate into product in 1 minute. B. Specific activity of an enzyme preparation is expressed as cat per kilogram or U per milligram of protein. C. Molar activity of an enzyme is cat per mole of the enzyme. D. Turnover number of the enzyme is the number of molecules of substrate transformed per catalytic site of the enzyme per minute. After understanding the mechanism of enzyme action, we move on to enzyme kinetics. What is enzyme kinetics? Let's find out. 4.6 Enzyme kinetics. The study of the rate at which an enzyme works is called enzyme kinetics. You have read in the last section that L. Michaelis and M. L. Menton developed a general theory of enzyme action and kinetics in 1913. According to this theory, we learned that in an enzyme-substrate reaction, the enzyme E first combines with the substrate S with the formation of an enzyme-substrate S complex, which subsequently breaks down into the product P, and the enzyme E is recovered. The reactions are assumed to be reversible, and K1, K2, K3 and K4 are the rate constants of each reaction. Let us examine enzyme kinetics as a function of the concentration of substrate available to the enzyme. The enzyme-substrate interaction may be studied with the help of figure 4.5. In this graph, substrate concentration and velocity of reaction of the enzyme catalysis has been shown on x and y axis, respectively. When the velocity of a reaction is plotted against different substrate concentrations, as in the present graph, a hyperbolic curve is obtained. Figure 4.5, effect of substrate concentration on the reaction velocity of an enzyme-catalyzed reaction M, N and O are 3 points on the curve representing 3 stages of enzyme-catalyzed reaction. While M represents that stage of the reaction when the substrate concentration is very low and the rate of reaction is directly proportional to the substrate concentration, O represents the stage when the reaction velocity reaches its maximum due to the gradual increase in substrate concentration resulting in a saturation of the active site of the enzyme. In between these two extremities lies the third point N, which represents the reaction velocity of the enzyme-catalyzed reaction, that is half of the maximum velocity. The mathematical relationship between the initial velocity of an enzyme-catalyzed reaction, the concentration of the substrate and certain characteristics of the enzyme are expressed by the Michaelis-Menten equation, which was derived on the basis of Michaelis-Menten theory and assumptions of G. E. Briggs and J. B. S. Haldane, and was proposed in 1925. The Michaelis-Menten equation is V equals V max into S upon Km plus S, where V is the velocity of the reaction at any stage, V max is the maximum velocity, S is the substrate concentration, and Km is the Michaelis-Menten constant, usually expressed in moles per liter. Km represents the substrate concentration at which the velocity of the reaction is half of V max. Km is, roughly, an inverse measure of the affinity or strength of binding between the enzyme and its substrate. The lower the Km, the greater the affinity, so, lower is the concentration of substrate needed to achieve a given rate. This equation is widely used to describe the most enzyme-catalyzed reactions. Now, we can verify the situations of the 3 points M, N and O in figure 4.5 with the help of this equation. Point M. At this stage of enzyme-substrate reaction, the substrate concentration is much less than Km and its value does not changes significantly with the increase in substrate concentration. Thus, S can be ignored in the denominator of the Michaelis-Menten equation. So it becomes V equals Vmax into S upon Km, as Vmax and Km are both constants. Vmax per kilometer ratio may be replaced by a new constant, K. Thus, we have SOCE, reaction velocity, V, of the enzyme-substrate reaction at this stage is directly proportional to substrate concentration, S. Point N. At this point, substrate concentration, S, is equal to Michaelis-Menten constant, Km. Thus, the Michaelis-Menten equation becomes V equals Vmax into S upon S plus SORV equals Vmax into S upon 2 SORV equals Vmax upon 2. This clearly established that Km is the substrate concentration at which the reaction velocity of an enzyme-catalyzed reaction is half of maximum velocity. Point O. At this point, the substrate concentration is much higher in comparison to Km, and so Km may be ignored in the Michaelis-Menten equation. Thus it becomes V equals Vmax into S upon SORV equals Vmax. Plotting the reciprocals of the same data points yields a double reciprocal or Lineweaver-Burke plot. This provides a more precise way to determine Vmax and Km. Let us get to learn more about this concept. Lineweaver-Burke equation, Michaelis-Menten equation, is often algebraically transformed to other forms for convenience in plotting experimental data. One such form is Lineweaver-Burke equation, which is derived by taking the reciprocal of both sides of Michaelis-Menten equation. This results 1 upon V equals Km plus SUPON Vmax into SOR 1 upon V equals Km upon Vmax into S plus SUPON Vmax into S. Now, after rearrangement we get 1 upon V equals Km upon Vmax into 1 upon S plus 1 upon Vmax. This is known as Lineweaver-Burke equation. Look at figure 4.6. When 1 V is plotted against 1 S, a straight line is obtained having a slope of Km, Vmax, an intercept of 1 Vmax on the 1 V axis and an intercept of 1 per kilometer on the 1 S axis. Figure 4.6, Lineweaver-Burke plot. Note that Km is not a fixed value but may vary with the structure of the substrate, pH and temperature. For enzymes having more than one substrate, each substrate has a characteristic Km. Affinity of an enzyme for a substrate inversely varies with the Km. For example, glucose can be acted upon by both hexokinase and glucokinase, but hexokinase has lower Km value for glucose and therefore greater affinity in comparison to glucokinase. At this stage, we suggest you take a break. Try to recapitulate what you have learned so far. We agree this section may have been a bit tough. Perhaps give this section another reading. Alternatively, try to attempt the check your progress exercises given next. This will help you clarify your doubts and facilitate your understanding about the topic. 4.7 Factors affecting enzyme activity. Rate of an enzyme-substrate reaction depends on several important factors. These include concentration of enzyme, concentration of substrate, pH, temperature, etc. Without the optimum condition of these factors, the enzymes will be unable to exhibit its best activity. These factors are discussed briefly in this subsection. Concentration of enzyme. Increase in enzyme concentration will increase the rate of an enzyme-catalyzed reaction. This is because of the availability of additional catalytic sites to which the substrates can bind. Accordingly, a straight line graph is obtained as can be seen in figure 4.7. Since the concentration of substrate relative to that of enzyme is always very high, increase in enzyme concentration results in increased enzyme activity. Concentration of substrate. When the concentration of the substrate is low, the rate of enzyme-catalyzed reaction also remains low, in spite of concentration of substrate being higher compared to enzyme concentration. This is because at this stage all the catalytic sites of the enzyme are not occupied by the substrate molecules. So, with the increase in substrate concentration, the rate of the reaction also increases until all the catalytic sites of the enzyme are utilized. Rate of the reaction becomes maximum at this point, and beyond this, it remains constant. Temperature. The rise in temperature accelerates the rate of enzyme-catalyzed reaction up to a certain temperature known as optimum temperature for the enzyme. At very high temperature, the enzyme undergoes denaturation and subsequent loss of activity. For most enzymes, the optimum temperatures are close to that of ambient temperature of the cell. For human beings, the temperature is in the region of 37 oC, while the optimum temperature of plant urease is 60 oC. Certain microbial enzymes have a higher optimum temperature that enables them to adjust in a new higher ambient temperature of a new environment. The effect of temperature on the reaction rate is almost zero at 0 oC, and this gradually increases with the rise of temperature until the optimum point reaches. Beyond this point, the activity of the enzyme falls due to denaturation and the curve bends, reaching ultimately the zero level. It has been found that a rise of 10 oC will double the activity of the enzyme. pH enzymes are influenced by pH changes as they have a ionic character due to the presence of amino and carboxylic groups. All enzymes have an optimum pH for showing highest catalytic activity, and a change of this affects their activity. However, within a narrow pH range, the changes in the reaction rate are reversible, but if the pH becomes too low or high, denaturation of the enzyme may occur. Most enzymes have an optimum pH range between 5 and 8, although some enzymes like pepsin and trypsin are most active at high acidic pH 1.5 and high alkaline pH 8 condition. A typical curve of enzyme activity against pH changes. Activators. Activity of many enzymes is influenced by certain icons called as activators. Large number of enzymes such as hexakinase that require ATP are also in need of divalent cations like Mg2+ or Mn2+. Many enzymes such as ATPase require monovalent cations like Na+, K+, or NH4+ for maximum catalytic activity. Amylase requires Cl. Generally, these icons interact with the substrates so that the substrates can bind with the catalytic sites of the enzyme properly. Thus, in absence of the activators, the enzymes become inactive or sluggish. Oxidation. Some enzymes which have the sulfhydryl L-SH group in the catalytic site are very sensitive to oxidation. Due to oxidation of the SH group by aerial oxygen or oxidizing agents, a disulfide linkage, SS, forms with the subsequent loss of enzyme activity. The enzyme activity can be restored by the reduction of the enzyme by some reducing agent such as cysteine or glutathione. Having studied about the factors influencing the enzyme activity, we move on to factors which inhibit the enzyme activity in the next section. 4.8 Enzyme inhibition. Enzymes are often inhibited by the presence of suitable inhibitors. Much of current drug therapy is based on this. Basically, there are three major classes of enzyme inhibition. These are competitive inhibition, when the substrate and inhibitor compete for binding to the same active site; non-competitive inhibition, when the inhibitor binds somewhere else on the enzyme molecule, reducing its efficiency; and uncompetitive inhibition. Let us learn about each of these classes of inhibition. Competitive inhibition. This type of inhibition takes place when a compound having a strong structural resemblance to the substrate competes with it for the catalytic site of the enzyme. Once the compound binds, the enzyme cannot convert the inhibitor to products. Increasing substrate concentration, however, is capable of displacing the inhibitor. This type of inhibition is reversible in nature. A good example of this is the reaction catalyzed by succinate dehydrogenase in the citric acid cycle. You will learn about the citric acid cycle in Unit 6 later in this course. In this reaction, succinate is converted to fumarate with the aid of this enzyme. Now, the compound malonate is structurally similar to succinate, and if present, it will compete with succinate for the catalytic site of succinate dehydrogenase and reduce the product formation. Thus, malonate is a competitive inhibitor for this particular reaction. In this type of inhibition, the Km of the enzyme for the substrate shows an apparent increase in the presence of the inhibitor, as can be seen in figure 4.12. This means that by increasing the substrate concentration, enzyme inhibition can be overcome. However, the Vmax remains unaltered. Figure 4.12, Lineweaver-Burke plot for an enzyme-substrate reaction in competitive inhibition. Next, let us learn about the non-competitive inhibition. Non-competitive inhibition. In this type of inhibition, the inhibitor binds at a site on the enzyme other than catalytic site. As there is no competition between the substrate and the inhibitor, the inhibition cannot be reversed in this case by increasing the substrate concentration. It appears that as if inhibitor is removing the enzyme, thus causing a decrease in Vmax, as can be seen in figure 4.13. No change of Km, however, occurs. Figure 4.13, Lineweaver-Burke plot for an enzyme-substrate reaction in non-competitive inhibition. A non-competitive inhibitor can combine with either the free enzyme or the enzyme-substrate complex, interfering both. The most common type of non-competitive inhibition is affected by the substances that combine with some functional group of the enzyme outside the catalytic site, that is essential for maintaining the conformation of the enzyme molecule required for its activity. For example, enzymes possessing the essential SH group are sometimes inhibited by metals like mercury or copper. Finally, let us get to know about the third type of enzyme inhibition, i.e. uncompetitive inhibition. Uncompetitive inhibition. In this type of inhibition, the inhibitor only binds with the enzyme-substrate complex, making it inactive. As a result, the product formation becomes difficult. In uncompetitive inhibition, both Km and Vmax changes, as can be seen in figure 4.14. The former increases while the latter decreases. This kind of inhibition is rare in one substrate reactions but common in two substrate reactions. Figure 4.14, Lineweaver-Burke plot for an enzyme-substrate reaction in uncompetitive inhibition. So far we studied about the enzyme mechanism and the factors which influence and inhibit its activity. Our study of enzymes shall be incomplete without the understanding of the role of enzymes and coenzymes in metabolism. The next section focuses on this aspect. But before moving on to the next section, let us recapitulate what we learned till now. 4.9 Role of enzymes and coenzymes in metabolism. You have already learned that many enzymes require a non-protein part for their optimal activity, which may be a coenzyme or a metal ion. This has also been stated earlier that many water-soluble vitamins are acting as coenzymes in their native or derived forms. So, it is important to learn how the coenzymes are assisting different enzymes in catalyzing biochemical reactions. In this subsection, you will learn about the chemical nature of different coenzymes and the general role of coenzymes in assisting different enzymes during metabolism. As coenzymes participate in a variety of functions, they can be classified broadly into two groups, I. Hydrogen transferring coenzymes and E. Group transferring coenzymes. Let's learn about these two groups of enzymes. I. Hydrogen transferring coenzymes. This group consists of three important coenzymes, all of which assist different enzymes in oxidation-reduction reactions. These are A. Nicotinamide nucleotides, B. Flavin nucleotides, C. Lipoic acid. A brief review of these coenzymes follows. A. Nicotinamide nucleotides. These coenzymes are derived from the vitamin niacin. You may recall reading in Unit 3 that they are of two types, nicotinamide adenine dinucleotide, NAD+, and nicotinamide adenine dinucleotide phosphate, NADP+. Collectively, they are called as pyridine nucleotides. Have a look at figure 4.15. In NAD+, the pyridine ring is attached to a ribose molecule through glycosidic bond, and the phosphate provides a link between adenosine and nicotinamide riboside. In NADP+, an additional phosphate group is present in carbon atom 2 of the ribose molecule of adenosine component. Oxidized and reduced forms of NAD and NADP. The pyridine ring of NAD+ is reduced by addition of a hydride ion to C4 when NAD+ is converted to NAD, and when NADP+ is converted to NADPH. In NADP, 2-hydroxyl group of the sugar ring of adenosine is phosphorylated. Figure 4.15, oxidized and reduced forms of NAD and NADP, both NAD+ and NADP+ act as hydrogen acceptors during oxidation-reduction reactions in the body. Dehydrogenation is the primary mechanism of biological oxidation in which two hydrogen atoms are removed from the substrate in presence of an acceptor. The hydrogen atoms ionize to yield 2H+ and 2 electrons. The nicotinamide ring of NAD+ or NADP+ accepts a proton and 2 electrons which are equivalent to H. The other H+ remains as such. All reactions catalyzed by them are reversible. Table 4.3 lists some of the enzymes, all dehydrogenases, which are dependent on these coenzymes. Table 4.3, enzymes requiring nicotinamide nucleotide coenzymes, B. Flavin nucleotides are derived from the vitamin B2, riboflavin, and are actively involved in hydrogen transfer reactions. You may recall reading in the last unit about the two coenzymes that are produced from riboflavin, i.e. flavin mononucleotide, FMN, and flavin adenine dinucleotide, FAD. Look at figure 4.16 for the structure of FAD. FMN is the active component of riboflavin and is formed by the addition of a phosphate group, and FAD is formed by the combination of FMN with one molecule of adenosine triphosphate, ATP. Both FMN and FAD are involved in many oxidation-reduction reactions. Enzymes that require the presence of FMN for their catalytic activity include glycolic acid oxidase, L-amino oxidase, etc. Fat-containing enzymes are succinate dehydrogenase, D-amino oxidase, etc. Figure 4.16, structures of riboflavin and its coenzyme FAD, C. Lipoic acid acts in the transfer of hydrogen during oxidative decarboxylation reactions. It occurs both in oxidized and reduced forms. Refer figure 4.17a for its structure. Figure 4.17b shows the structure of lipoamide, where lipoic acid is bound in an amide linkage to the epsilon amino group of a lysine residue, blue, of dehydrolipoamide acyl transferase. The complex reactions of the carbohydrate metabolism catalyzed by pyruvate dehydrogenase system and alpha-ketoglutarate dehydrogenase system require the participation of lipoic acid. These reactions will be discussed later in detail in Unit 6 covering carbohydrate metabolism. Figure 4.17, structures of A. Lipoic acid, B. Lipoamide 2, group transferring coenzymes. This group consists of the following coenzymes involved in different metabolic reactions, where the transfer of any functional group is taking place: biotin, thiamine diphosphate, TDP, pyridoxal phosphate, coenzyme A, CoA, tetrahydrofolic acid, THF, cobamide coenzymes, adenosine triphosphate, ATP. You may recall reading about these enzymes in the last unit on vitamins, particularly under the section on water-soluble vitamins. There we talked about them as derivatives of water-soluble vitamins. We shall look at these enzymes once again to help you understand them better. Biotin. This water-soluble B group vitamin participates in the transfer of carboxylic groups. Two important enzymes carrying out carboxylation reactions are pyruvate carboxylase and acetyl-CoA carboxylase. Pyruvate carboxylase contains 4 biotin molecules attached to the enzyme protein. The biotin-dependent enzymes require ATP, adenosine triphosphate, which is converted to ADP, adenosine diphosphate, during the reaction. Figure 4.18, structure of biotin and enzyme-bound biotin. Thiamine diphosphate, TDP, is the coenzyme responsible for the transfer of aldehyde and glyoxyl groups. Figure 4.19 presents the structure of TDP. Figure 4.19 Structure of thiamine diphosphate. The most important metabolic reaction catalyzed by TDP is the oxidative decarboxylation of alpha-keto acids such as pyruvic acid. The involvement of TDP in the decarboxylation of pyruvic acid was so well known that the coenzyme was also popularly called as co-carboxylase. It is also actively taking part in the conversion of pyruvic acid to acetyl-CoA and alpha-ketoglutarate to succinyl-CoA that are catalyzed by pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase complex, respectively during carbohydrate metabolism. You shall learn about this in more details in Unit 6. Another important metabolic reaction catalyzed by TDP is the conversion of XYLULOSE5-phosphate to SEDOHEPTULOSE7-phosphate catalyzed by the enzyme transcatolase in pentose phosphate pathway of carbohydrate metabolism. Look up Unit 6, Section 6.8 for knowing more about the pentose phosphate pathway. Pyridoxal phosphate. Pyridoxal phosphate is derived from pyridoxine, vitamin B6, and is involved in amino acid metabolism. The other two compounds, pyridoxal and pyridoxamine, about which you learnt in the last unit, having the properties of vitamin B6 also occur as phosphate derivatives. Enzymes that are dependent on B6-phosphate coenzymes catalyze a variety of reactions such as transamination, transfer of amino group from an amino acid to a keto acid, decarboxylation, removal of carboxyl group, and racemization, transformation of one isomer to another. Figure 4.20 Structure of pyridoxal phosphate, PI, vitamin B6, enzyme glutamate oxaloacetate transaminase catalyzes the reaction between glutamate and oxaloacetate with the formation of alpha-ketoglutarate and aspartate due to transfer of one amino group from glutamate to oxaloacetate. Pyridoxal phosphate acts as the carrier of the amino group. Whenever it accepts the amino group, it transforms to pyridoxamine phosphate, and after the release of the amino group, it again becomes pyridoxal phosphate. Aspartate is decarboxylated by aspartate decarboxylase taking the help of pyridoxal phosphate. L-alanine transforms to D-alanine by alanine racemase, which also requires pyridoxal phosphate as the coenzyme. Coenzyme A is derived from the vitamin pantothenic acid. This is abbreviated as CoA. This can be divided into two components, adenosine 3,5-diphosphate and pantothene, which is formed by the combination of pantothenic acid and mercaptoethylamine. Refer Figure 4.21 to understand its structures well. It gives rise to acyl-CoA derivatives that are mainly formed in ATP-dependent synthetase reactions. These are highly reactive and participate in various types of reactions. For example, oxaloacetate is converted to citrate in presence of citrate synthetase by accepting acyl group of acyl-CoA. During carbohydrate metabolism, the pyruvate is converted by the pyruvate dehydrogenase complex to acetyl-CoA by the active participation of coenzyme A. Also, in the oxidation of fatty acids, CoA has an important role to play, as you will find out in Unit 7. Figure 4.21 Structure of coenzyme A. Tetrahydrofolate. Coenzyme tetrahydrofolate, as you may already know, is derived from the vitamin folic acid. Have a look at the figure 4.22 for structures of folic acid and tetrahydrofolate, the coenzyme. It is responsible for the transfer of one carbon fragments at the oxidation level of formate, formaldehyde and methanol. The two most important metabolic reactions in which tetrahydrofolate participates are the biosynthesis of purine, refer to Unit 8, subsection 8.3.1, and methionine. Figure 4.22 Structures of folic acid and tetrahydrofolic acid, cobamide coenzymes. Cobamide coenzymes are the derivatives of vitamin B12. The structure of cobamide coenzyme is very complex and is shown in Figure 4.23. They participate in many biochemical reactions. L-methylmalonyl-CoA is converted to succinyl-CoA by the action of the enzyme methylmalonyl-CoA isomerase. In this reaction, the COS-CoA group is transferred by the cobamide coenzyme. An important reaction catalyzed by this coenzyme as an integral part of the ribonucleotide reductase is the reduction of ribonucleotides to deoxyribonucleotides. Figure 4.23, structure of vitamin B12 and its coenzyme, adenosine triphosphate. Though adenosine triphosphate, ATP, often functions as a second substrate, it can also serve as a coenzyme by modulating the activity of specific enzymes. The compound consists of adenine connected to a ribosyl 5' triphosphate. See figure 4.24 for its structure. As a co-substrate, ATP is utilized by various kinases for the transfer of the terminal phosphate group to various acceptors. For example, glucose is converted to glucose 6-phosphate in presence of the enzyme hexokinase by accepting phosphate from ATP. This will be discussed many a times in the Unit 6 and 7 covering carbohydrate and lipid metabolism. Figure 4.24, structure of ATP. So far we have studied about the different enzymes and coenzyme involved in metabolism. Interestingly, it is known that enzymes catalyzing essentially the same reaction may differ in various ways. These are called isozymes. The next section focuses on isozymes. 4.10 Isozymes. Sometimes an enzyme present in the same organism is found to have different molecular forms but catalyzing the same reaction. These are called isozymes. The 1964 committee recommended that multiple enzyme forms in a single species should be known as isozymes. Among many enzymes known to have isozymes, the most studied is lactate dehydrogenase, LDH, which is an important enzyme of carbohydrate metabolism. This enzyme exists in 5 possible forms in most vertebrates. In fact, 2 basically different types of LDH occur predominantly in the heart and muscle. The former consist of 4 identical monomers, H. The latter, muscle LDH, also forms due to the combination of 4 identical monomers, M, but having different amino acid composition in comparison to the former. Different combinations of H and M monomers yield 3 additional hybrid enzymes possessing 4 monomers each. The possible combinations of H and M monomers therefore produce 5 isozymes of LDH. These are H4, H3M, H2M2, HM3 and M4. Isozymes differ from each other not only in the amino acid composition, they also have different electrophoretic property, thermolability, immunological properties and kinetic properties such as substrate affinities, i.e. different Km values. Estimation of isozymes of some enzymes can be used for the clinical diagnosis of different diseases. Let us learn about this aspect next. 4.11 Enzymes in clinical diagnosis. The rationale for measuring plasma or serum enzyme levels is based on the premise that these levels reflect changes that have occurred in a specific tissue or organ. Enzymes present in the blood are of 2 types. One type, such as thrombin, associated with blood coagulation, has a functional role and is present in high concentration. The other type has no functional role in the blood and is present in very small amount. The latter types of enzymes mainly originate from different tissues or organs. An insult in the form of any disease may cause changes in cell membrane permeability or increased cell death, resulting in the release of intracellular enzymes into the blood. As a result, the concentration of these enzymes increases in the blood. In the diagnosis of specific organ involvement in a disease process, it would be ideal if enzymes unique to each organ could be identified. But this seldom occurs as the metabolism of different organs is not unique. Alcohol dehydrogenase and acid phosphatase are 2 such enzymes, the levels of which in the blood can be used as specific tools for the diseases of liver and prostate, respectively. However, the ratio of enzymes varies from organ to organ. This fact, combined with a study of the kinetics of appearance and disappearance of particular enzymes from the blood, enables one to identify the involvement of a specific organ in a disease. Both glutamate oxaloacetate transaminase, GOT, and glutamate pyruvate transaminase, GPT, are present in the liver, but the concentration of GPT is more than GOT. In liver dysfunction, both these enzymes are leaked out from liver, and as a result, their level in the blood increases, but the rise is more in case of GPT than GOT, making it a reliable marker for liver diseases. Following a heart attack, many enzymes such as GOT, creatine phosphokinase, LDH etc., are released from the myocardium, but their time of release differs, enabling one to establish when the attack occurred and whether the treatment is effective or not. Enzymes that are often estimated in the plasma or serum for confirmatory or suggestive diagnosis of different diseases are listed in Table 4.4 along with their normal values. This has to be kept in mind that these values will vary according to the procedure used for their estimation, and generally all laboratories standardize their own normal values. This is to note that though increase of enzyme activities in the cerebrospinal fluid sometimes occurs, e.g. increase in lactate dehydrogenase activity in meningitis, it is not reflected in the blood. Isozymes are often estimated to specify a diseased organ, tissue. Two different isozymes of alkaline phosphatase are present in liver and bones. So, a rise of serum alkaline phosphatase may occur either due to liver damage or due to problems related to bones. In this situation, estimation of specific isozymes will give a clear picture for the rise in the enzyme level in serum, and this enables one to pinpoint the affected organ, tissue. Table 4.4 Common enzymes of diagnostic importance 4.12 Summary. In this we unit covered the structural and functional aspects of enzymes and coenzymes, two of the most important group of biomolecules. While enzymes are almost always protein in nature, coenzymes are mainly derived from water-soluble vitamins. You now have a clear idea about the different classes of enzymes and the type of reactions they catalyze. This knowledge will greatly help you to understand the different metabolic reactions that will be described in the subsequent sections. One of the main differences between an inorganic catalyst and enzyme is the high degree of specificity of the latter, which has been discussed in the light of models for enzyme-substrate interaction. Enzyme, being a protein, is particularly sensitive to different environmental factors and its activity changes accordingly. These, along with other important factors governing the activity of an enzyme, have been dealt with in this unit. Enzyme kinetics was further discussed in this unit, and so also the application of Michaelis-Menten equation. You now understand how the affinity of an enzyme for a particular substrate can be judged on the basis of its Km value. Impact of different inhibitors on the Km and Vmax of an enzyme is also taken into consideration. Further, in this unit we got the idea how different coenzymes are taking part in different enzyme-catalyzed reactions with suitable examples. This will also greatly help us in learning different metabolic reactions in the subsequent units of this course. Importance of serum enzymes and isozymes in the diagnosis of different diseases is well known. This has been discussed comprehensively. This unit has covered all important aspects of enzymology, but emphasis has been given only on the topics relevant to our need as nutritional biochemists. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.